The full title of Darwin's famous book is On the Origin of Species by Means of Natural Selection, or the Preservation of Favored Races in the Struggle for Life. Actually, in spite of the first part of this title, which is how we usually address the book, just On the Origin of Species, Darwin spent relatively little time addressing how he thought new species might actually arise. Instead, most of Darwin's effort was spent documenting the occurrence of evolution and providing evidence to support his theory of natural selection. Now, although he didn't solve what he called the species problem, Darwin did emphasize an important idea, and this was descent with modification, which suggested that all species living today somehow, or that ever lived, somehow descended from an original common ancestor that existed sometime in the very remote past. And this idea remains the dominant view of contemporary biologists, who generally agree that all life on Earth must have descended from a single ancestral species that lived over 3 billion years ago. Somehow, then, the evolutionary mechanisms we've discussed up to this point, such as natural selection or genetic drift, must sometimes lead to the creation of new distinct species. And this process, which we call speciation, must have occurred frequently, at least over a geological timescale, in order to account for the millions upon millions of different species that exist on the Earth. The fact that speciation occurs is well established, but how speciation occurs remains one of the most active areas of research in evolutionary biology today. In this lecture and in the next, we're going to look at what is known about how new species arise. As you might guess, the answer has something to do with biological information, essentially involving the separation and diversification of a single gene pool into two. In this sense, our discussion of speciation takes our analysis of biological information up yet another level in the hierarchy of life, from the level of populations to the level of distinctly different species. Well, the first thing we have to do is ask, what is a species? What do we mean by that? Now, the answer to this question may seem obvious at first, but it's more subtle than you might think. The word species literally comes from the Latin word which means kind or type. And when we look at organisms around us, they do often seem to fall into distinct kinds. Let me give you an example. I'm going to choose an example from birds. Okay? Now, if you take a walk in a typical field in the northeastern United States, you're likely to come across two very common types of small birds: song sparrows and chipping sparrows. These two kinds of birds live in more or less the same place. They're very similar in size, in shape, and in color, and at first glance, you might easily confuse the two if you just see them in a bush nearby. But even if you're not a bird watcher, with a pair of binoculars and a little bit of closer examination, you'd have no trouble distinguishing these two kinds of birds as different species. Although they're very similar, the two species are also distinctly different in a number of ways. For example, the song sparrow has distinct brown streaks on its breast, while the chipping sparrow has a reddish cap and bright white streaks on the side of its head. Now, even if you didn't know what to call these birds, you could always tell them apart. Humans have described organisms around them in this way since the earliest times of recorded history, and have separated them out and named them according to which ones appear more alike. Often the categories that are made this way correspond well to what biologists would want to consider as being distinct species. In the case of song and chipping sparrows, it works quite well. But the problem isn't always as easy as this. Consider the similarities and differences among a very different kind of group of animals. Think of a German shepherd and a wolf and a chihuahua and a beagle. Now, the German shepherd and the wolf look quite similar to each other, and they look much more similar to each other than either does to the chihuahua or the beagle. Yet we know that the German shepherd, the chihuahua, and the beagle are all the same species, just different varieties. And the wolf is a very distinct species. So in this case, differences in appearance alone or similarities aren't necessarily sufficient for us to define what is or isn't a species. The reverse is also true. Just because two organisms look very similar or even identical, they're not necessarily the same species. Here's another bird example. Willow flycatchers and alder flycatchers are also very common birds. They're found in the northeastern United States around wetlands. Like song sparrows and chipping sparrows, these two kinds of birds are often found in pretty much the same place. In fact, you can sometimes find one of each kind singing in the very same tree. Willow and alder flycatchers, I'll tell you, are considered to be distinct species. Yet these two kinds of birds look almost identical. If I caught a male of each species and held them side by side in my hands, even an experienced ornithologist couldn't tell them apart based just on how they look. The only way you can easily distinguish these two species is by the way they sing. They sing distinctly different songs. So, how can we reliably define a species in the face of confusing cases like this? It turns out that this relatively simple-sounding problem is one of the most important questions in evolutionary biology because it helps us understand the processes by which new species might be formed. The question doesn't have a simple answer, however. Now, the answer that most biologists agree comes closest to the mark in terms of answering the question, what is a species, was articulated in the 1940s by the Harvard biologist Ernst Mayr, who's one of the evolutionary theorists of the 20th century who made major contributions to the evolutionary synthesis that we talked about earlier. Mayr proposed what has become known as the biological species concept. The biological species concept defines a species as a population or a group of populations whose members have the potential to interbreed in nature and produce viable and fertile offspring, but who cannot successfully interbreed with members of other populations or other species outside that group. In other words, Mayr suggested that we define a species specifically in population genetics terms as the largest inclusive group of individuals among whom genetic exchange is possible. Or to put it a slightly different way, by Mayr's definition, a species is the highest level of organization in the hierarchy of living things at which biological information can be mixed. According to the biological species concept, then, what defines species and separates them as being distinct are barriers to the exchange of genetic information. The term we use for this is reproductive isolation. Now, the biological species concept handles both the dog-wolf example and the flycatcher example I mentioned a moment ago quite well. German shepherds can breed quite easily with beagles, but not with wolves, so the shepherd and the beagle are clearly the same species by this definition, while the wolf is distinct. Conversely, alder and willow flycatchers do not breed with each other, and so again, by the biological species concept, we can define them as being distinct species. Now, the biological species concept, unfortunately, does not account for a number of other kinds of cases we're interested in. It specifically has a difficult time dealing with special cases where what we think are distinct species might sometimes interbreed. And this does happen. I'm going to give you another bird example. Lazuli buntings and indigo buntings are very closely related, distinct species of birds. They look quite different from each other, and you'd have an easier time sorting these out than you would the sparrows I mentioned at the beginning. The indigo bunting, as its name suggests, is almost entirely a deep blue color, while the lazuli bunting has a blue head and back, but also has a very bright red breast and white belly and a distinct white bar on each wing. The songs of these species are also quite distinct. Furthermore, these species usually live in completely different parts of the country for most part. Indigo buntings east of the Mississippi River and lazuli buntings west of the Mississippi. So by any criteria that seems reasonable, we'd think these two kinds of birds should be considered two species. And as I said, they are. However, in the Midwestern part of the country, there is a region where the geographic distribution of these two species overlap, and you frequently find indigo and lazuli buntings living in the same area. And when they do occur in the same area, these birds will in fact mate with each other on occasion. They'll hybridize. And moreover, the offspring of these hybrid pairs will actually survive and potentially themselves reproduce. So in fact, if you go to the Midwest, you can see birds that look a little bit like a lazuli bunting and a little bit like an indigo bunting because they're truly morphological intermediates. There are many similar cases of distinct species that will occasionally hybridize in areas where their ranges overlap a little bit. Now, there are other problems with the biological species concept in terms of how we can apply it to the real world. And specifically, the biological species concept just simply can't be applied easily to organisms that are no longer alive, fossil species. which are of major interest to evolutionary biologists. We can't know if species that no longer exist would or wouldn't mate. Moreover, there are many species, including all prokaryotic organisms, that usually don't sexually reproduce at all. So we can't use the fact of sexual reproduction or not occurring between two groups of organisms to define them as species. Because of these limitations, biologists often use a number of other approaches to operationally define species, and I'm just going to mention two of these alternatives. The morphological species concept is really where we started when we were talking about the sparrows at the beginning of this lecture. This concept suggests that we should use morphological, identifiable morphological or otherwise measurable differences between groups of organisms as the best way to define species. Now, the underlying logic of this is the suggestion that these morphological differences in fact represent genetic differences between two groups of birds, and therefore it's compatible theoretically with the biological species concept, but it doesn't rely on the fact of reproductive isolation to distinguish species. And so the morphological species concept is actually widely used in practice because it can be applied in just about any case. The difficulty comes, however, with the kinds of confusing examples that I gave you at the beginning of this lecture. Now, the phylogenetic species concept is a newer approach based on recently developed methods for determining evolutionary lineages among groups of organisms, a set of methods referred to by the somewhat cumbersome name as phylogenetic systematics. Now, the logic of this is that how far away different groups of organisms have diverged from a common ancestor in evolutionary time might be used as a measure to help us decide whether or not those groups represent distinct species. In a couple of lectures, we'll talk more explicitly about phylogenetic systematics and how we might use them to determine historical evolutionary relationships. The take-home message for now, however, is that if we do know evolutionary histories of organisms in relationship to each other, we can use this information as an objective way to help define what is and isn't a species. Now, both the morphological species concept and the phylogenetic species concept have their own advantages and drawbacks. They can be widely applied to any kind of organisms. They don't rely on reproductive isolation for their use, although they both imply reproductive isolation is operating in the creation of species. They both have their drawbacks. We've already mentioned the drawbacks of the morphological species concept. It confuses cases of dissimilarities or similarities where we think that there practically should be species differences. The phylogenetic species concept also has the disadvantage of requiring a lot of information about the evolutionary history of a group of organisms to allow us to make inferences about similarities. And so it can't practically be applied to groups where that information just simply isn't available yet. In practice, biologists typically use a combination of different approaches to the problem of defining species depending on which group they're working with, the information available for that group, and the kind of question that they're asking. For some kinds of organisms, what is and isn't a species seems obvious, and there's no point in quibbling over the details about how we define them. In other cases, however, it's not so obvious whether organisms actually represent distinct species or not by any definition. Which brings us to the question of how species actually are formed. So let's turn our attention now to this process of speciation. If we take a close look at the fossil record, that is, if we trace the evolutionary lineage of living forms on the planet by examining the fossilized remains of related organisms that lived in previous times, we actually can see two different general patterns by which species appear. On the one hand, we find examples in which a series of fossils obtained across different geological time periods appears to form a linear progression from one species to another with a series of intermediates that display a steady range of small changes in shape, size, or other defining characteristics. We call this pattern anagenesis. And the implication of this word, ana, comes from the root meaning up, and genesis means origin. So this is essentially an origin of one form from the other through a continuous gradation. The new species that is formed here essentially has replaced a species that existed earlier in time. We don't get the formation of more than one new species, just an evolutionary transformation. through anagenesis. The implication behind this is that there is a gradual change reflecting the accumulation, gradual morphological change reflecting the accumulation of small genetic changes over time, which could be explained by the kinds of evolutionary forces we've discussed already, such as natural selection or genetic drift. Most of what Darwin was able to document and explain in his book On the Origin of Species involved examples of this sort of transformation of species over time through anagenesis. And in that sense, Darwin documented evolutionary change, but not the process by which one species might somehow split into two. Now, this is the other pattern of evolutionary transformation that we can observe in the fossil record, and we call it cladogenesis, which is also called branching evolution. The root clados means to branch or limb. Thus, this pattern of evolution reflects the process by which a single ancestral species somehow gives rise to two or more descendant species. Cladogenesis is of special interest to us at this point because it represents the splitting in two of one species. And this is what we want to focus on for the rest of today's lecture. Okay, to start this discussion, let's go back to one of our examples from last time, the black-bellied seed cracker. You remember, that's the bird that lives in Cameroon in West Africa. And it's characterized by having a distinct bimodal distribution of beak sizes. Now, given that this bird has such a strong dimorphism in its beak size, we asked, why are these birds considered two species or one? And in fact, these birds are considered just a single species. Why do we consider these one species and not two, even though there is this obvious morphological difference between large-billed and small-billed birds? The reason is simply this. They're considered a single species because all individuals in the population will mate with one another irrespective of beak size. That is, a large-billed male will just as readily mate with a small-billed bird, a female, as it will with a large-billed female, and vice versa. Furthermore, remember from our earlier discussion that birds with intermediate beak sizes are born into the population. This makes sense since beak size is a continuous character and because birds with different beak sizes will freely mate. It's just that the birds with these intermediate beak sizes are selected against when they're young and so don't survive to adulthood. Well, that led us to another question. Given this strong disruptive selection, is it possible that these birds may someday evolve into two species? Or to put it another way, what would be necessary? What would it take for the two morphs to become two separate species? The answer to this question is that the two species could diverge into the two subpopulations of large-billed and small-billed birds could diverge into two distinct species only if they stopped mating with each other. Why is that the necessary condition? Because as long as there are two morphs that interbreed, they exchange genetic material. That is, there's gene flow between the subpopulation of large-beaked and small-beaked birds. If, on the other hand, the large-beaked and small-beaked birds stopped mating with each other, two distinct gene pools could be created, and these gene pools could begin to diverge from one another. And then we might expect that the pattern of selection we observe here, disruptive selection, to eventually lead to two distinct species. There would no longer be intermediates formed because there would no longer be breeding between the two extremes. This example brings us to the essential requirement for speciation, and that is that speciation results as the outcome of the separation or isolation and subsequent divergence of gene pools. Once gene pools have become isolated, then the evolutionary mechanisms we've already discussed, notably mutation, drift, and selection, can modify those independent gene pools in different ways, resulting in diverse species with diverse adaptations. But this conclusion begs another question, which is really the gist here. And the question is this: what would cause these two subpopulations to become reproductively isolated? Why would they stop mating? This really is the important question because it's the most critical step in the formation of new species, the step of initially separating the gene pool of an ancestral species into two isolated gene pools. Something has to happen that will step in and prevent gene flow from occurring. Well, there are thought to be two main sets of conditions under which gene flow can be interrupted in this way. These conditions correspond to what we generally refer to as two main modes of speciation. So-called allopatric speciation occurs when one population becomes physically divided in two by a physical barrier, creating geographically isolated subpopulations. Allo means different, patris means country. So allopatric means living in a different place. When organisms are found in different places, we say they're allopatric. So allopatric speciation refers to the case where new species arise in different places. By contrast, sympatric speciation occurs when new species diverge from a common ancestor without any geographic separation. That is, without the subpopulations that diverge from ever being physically separated from each other. The root sim means same, so we say that organisms are sympatric when they live in the same place. The way in which gene flow can be interrupted when subpopulations remain in close contact is harder to envision. And until recently, some evolutionary biologists were skeptical that sympatric speciation occurred at all. We're going to look at allopatric speciation for the remainder of this lecture, and then continue our discussion next time by looking at mechanisms underlying sympatric speciation. Now, the fundamental mechanism thought to be responsible for allopatric speciation is actually relatively straightforward. Let's begin to consider this by imagining a population of some species that has a relatively broad geographic range. Now imagine that some physical barrier arises that divides this population into, for example, let's say we're talking about a species of plant that lives on a broad plain between two mountain ranges, and let's say that sea levels rise. This happens frequently in evolutionary history. The lower elevations in the center of this broad plain would become flooded, in our hypothetical example, permanently, making it so that our hypothetical species can now only exist in the highlands on the two sides of what was once a continuous distribution across the plain. So what was once one large continuous population would be separated in this way into two geographically isolated subpopulations, with the sea that's flooded the center being an insurmountable barrier, preventing the dispersal of individuals from one side to the other, and thus preventing individuals from one side from mating with individuals from the other. Now, the example I'm developing here is a hypothetical one, but the appearance of geographic barriers like this that divide a species range in two is really quite common in geological history. Examples include the uplifting of mountain ranges, the appearance of geographic barriers like canyons, the movements of glaciers, and even on the longest time scale, continental drift. When a physical barrier divides what was a previously geographically isolated population like this, we refer to it as a vicariance event. And we refer to the resulting disjunct distribution of what was once a continuous population as a vicariance distribution. Now, another way in which a subpopulation might become geographically isolated from the rest of its species occurs when some number of individuals disperse across an existing barrier and found a new geographically isolated population. In this case, we call the disjunct distribution that results a dispersal distribution. The Darwin's finches we've referred to in a couple of contexts now provide an example of an island radiation of species, with radiation here meaning the evolution of a large number of species from a single ancestor. And this radiation is thought to have arisen from the geographic separation of a founding population that initially colonized one of the Galapagos Islands and then subsequently would colonize the remaining islands. Each island then had its own geographically isolated population. Gene flow was prevented from occurring between these isolated populations and the original population, presumably from the mainland where the original birds came. And so over time, we would have distinctly different gene pools on each island. Now, when populations become separated by a geographic barrier, it's often the case that the two populations will be living in slightly different environments, perhaps very different environments. For example, there may be different amounts of rainfall, different average temperatures, different soil qualities, and so forth in the hypothetical example of plants that we've been talking about. To the extent that geographically isolated populations experience different environmental conditions, we expect natural selection to act differently on these two populations, with each becoming adapted in a different way to its own specific environment. Selection, then, will cause gene pools of these subpopulations to diverge away from each other over time. Now, depending on population size, we would also expect genetic drift to act on gene pools of two isolated subpopulations, leading to genetic divergence even in the absence of selection. Genetic drift is probably especially important in the case of colonizing events or dispersal distributions that I mentioned earlier, such as the island radiation of Darwin's finches, because colonizing populations tend to be very small. These founding populations then go through what we called earlier a genetic bottleneck, and drift can become an overwhelming and dominant force affecting the allele frequencies in those populations. As the result of natural selection, or genetic drift, or both, the gene pools of geographically isolated populations eventually may diverge to such a large extent that individuals from one of the separated populations may no longer be able to successfully mate and produce offspring with individuals from the other population. That is, as a happenstance of the divergence that's occurred between the two gene pools, something might happen to aspects of the individual's behavior or reproductive physiology or some other aspect of their biology that just simply makes it unlikely or impossible for those individuals to ever mate again. Individuals from the two populations successfully. This is interesting because what it means is that for some reason those two populations join up together at some future point in evolutionary time, their gene pools remain isolated. We say that they've become reproductively isolated. Let's go back to our plant example that we started with. Let's say the sea level has dropped again at some later point in geological time. The plants that were inhabiting the two subpopulations on the highlands on either side of the sea that had been created can now begin to slowly extend their range back into the center of what was once the continuous distribution. And eventually those two plant populations will start to overlap physically. The geographic isolation that once existed now has disappeared. But if those two plant populations have diverged enough genetically because of selection or drift when they were in geographic isolation, then it's possible that for a number of reasons they'll no longer be able to produce offspring. They've become reproductively isolated, and by the biological species concept, we would say that they've become two different species. Now, I said earlier that speciation is the outcome of isolation and divergence of gene pools. In allopatric speciation, gene flow between two subpopulations is initially blocked by a geographic barrier, and it's not too hard to imagine how this can occur. In a sense, allopatric speciation is a very intuitive concept. And in fact, it's thought to occur frequently. So most evolutionary biologists would suggest that allopatric speciation is the most common way over the evolutionary history of the Earth that new species have arisen, simply because for whatever reason they become separated in space for some period of time. But what about sympatric speciation? How can the gene flow between two subpopulations of a single species initially become blocked if those two subpopulations are not separated in any physical sense? This is the question that we're going to pick up with in the next lecture.